Hello everyone and welcome back to Creation Myths. Today I have what I think is a really fun video. In this video we're going to talk about how I blundered. This is a response to Dr. Rob Carter from Creation Ministries International, and there's a bit of a plot twist with this one because folks, let me tell you, by the end of this video you're going to see Dr. Carter making the same argument I'm making. By the end of this video, Dr. Carter is going to agree with me. So let's get into what's going on here. This video is my turn in a bit of a game of ping pong that Dr. Carter and I have been playing since he participated in this live stream on January 8th, 2025. Now, in this live stream, he gave a presentation and then did a Q&A, and in particular during the Q&A, he said some really notable things with implications for the young Earth creationist calculations for the time to the mitochondrial and Y chromosome most recent common ancestors. That's mitochondrial Eve and Y chromosome Adam, even though really it should be Y chromosome Noah, but everyone just calls it Y chromosome Adam. Whatever, it doesn't matter. So anyway, Dr. Carter said some really interesting stuff in here that a couple people watching at the time thought was notable, and he answered a question in a way that made it, I think, even more notable. So I pointed this out in this video. Professional Creationist makes a huge admission because what he said and what he described in his answer to the question in that interview, in the Q&A to that presentation he did, was how you cannot assume that there is just strictly neutral accumulation of mutations in the human genome, right? There's other stuff going on that disrupts that neutral rate of accumulation. And specifically, he went out of his way to say that most of the variation is lost, and he described conditions under which it's not just neutral genetic drift that is responsible for that loss of variation. That's important because in order for the short-term mutation rate to match the long-term substitution rate, the rate at which mutations accumulate in lineages, you need to have conditions of strict neutrality. Once you have things like non-random mating or natural selection, it throws that math off and those two numbers start to diverge from each other. And that matters because that is one of the most important young Earth creationist arguments. The argument that those two rates, the mutation rate and the long-term substitution rate, are actually equal. Because in order to get the calculations, in order to get that time to the most recent common ancestor to fit in the young Earth timeline, 4,500 years, give or take, for the Y chromosome and approximately 6,000 years for the mitochondrial DNA, in order for that to work, you need those two rates to be equal, which means you can't acknowledge that things like natural selection are operating on those parts of the genome. Dr. Rob Carter responded with this video, and it's this to which I am responding right here. Now, this video was long. It was like an hour and 20 minutes long, so I'm not going to cover most of it. I'm going to go through the basic argument that Dr. Carter made, and we're going to talk about the important parts and where he's right and where he's wrong. But this is not the end of the story, because since this video came out and I started working on this one, Dr. Carter released another video on this topic. It's this video. The saga of mitochondrial Eve continues, and as you can see in the thumbnail, massive update. This video is going to be super important when we get around to the end today, because this video right here, this is where Rob Carter agrees with me. I don't know if he realizes that's what he did, but the words he says in this video, the thing he describes, is the same thing that I am saying. So by the time we get to the end here, as of this recording in Dr. Rob Carter's most recent video on the time to most recent common ancestor for mitochondrial Eve and mutation rates and substitution rates, as of this recording, Dr. Carter and I agree on the processes that drive the evolution of the mitochondrial DNA, and therefore we agree on whether the mutation rate and the substitution rate are the same. And the answer is that they are not. So I'm going to start here by summarizing Dr. Carter's argument in that long video that I just described, the one that's about an hour and 20 minutes long. I'm not going to go through and show a bunch of clips because this video that we're doing right now, this is going to be long enough as it is. So Dr. Carter, when you see this, I'm assuming you're going to see this, when you see this, if you think that anything here is misrepresenting anything you say, please let me know and I'd be happy to correct my errors. But I think this is an accurate representation of the argument that you made. So he says that he didn't contradict Dr. Jeanson in his answer during the Q&A after his presentation on that live stream from January of 2025. He says that he and Dr. Jeanson were talking about different parts of the genome. Jeanson focuses on the mitochondrial DNA and the Y chromosome, and Dr. Carter was talking more broadly about the autosomal DNA, the nuclear DNA excluding the sex chromosomes and specifically excluding the Y chromosome. Now, specifically with the non-recombining mitochondrial DNA in the Y chromosome, they are haploid and they do not experience homologous recombination like the rest of the genome. Therefore, because of that, they accumulate mutations in a ratchet-like fashion. And I think Dr. Carter here several times is alluding to the concept of Muller's ratchet without actually saying it, but I think that's kind of the concept he's alluding to when he's describing mutation accumulation as ratchet-like with regard to the mitochondrial DNA and the Y chromosome. And then finally, he says that the majority of mutations are selectively neutral and therefore accumulate at the neutral rate, i.e. mutation rate equals substitution rate, or in other words, the single generation pedigree mutation rate equals the phylogenetic mutation rate, the rate at which mutations accumulate in evolutionary lineages over time. Again, I think this is a fair representation of Dr. Carter's argument in that video that was a direct response to me. I'm really striving here to make sure that I am accurately portraying his argument before I show why it's wrong. On Dr. Carter's first point about whether or not he contradicted Dr. Nathaniel Jeanson, Dr. Carter says that he was talking about autosomes, or more properly, the diploid parts of the human genome. And what he said does not apply to the mitochondrial DNA and the Y chromosome, which is what Dr. Jeanson's calculations are with regard to. So he wasn't contradicting Dr. Jeanson, and in fact that he and Dr. Jeanson agree on the stuff that he's talking about. Now, you can go back and listen if you want, but I think it's clear from the question he was answering that both the questioner, the person who asked the question, and Dr. Carter in his answer were referring to the common young Earth time to most recent common ancestor calculations. There was actually a funny moment in that live stream that I showed in my previous video in this saga where he assumed that the person asking the question was me. And I only ever talk about this stuff in the context of the mitochondrial DNA and the Y chromosome. The fact that he thought it was me tells me that he knew we were talking about the mitochondrial DNA, the Y chromosome, and all this mutation rate, substitution rate stuff that's related to Dr. Jeanson's publications and calculations. So, I don't think there's any ambiguity about the context and the content of what was being talked about there. I think this response that they were talking about two different things is a bit of face-saving. But I'm not going to belabor the point. He spends the rest of that video making the case for we can call them the Jeanson rates, if you will, right? Using the mutation rate as the substitution rate as Dr. Jeanson specifically describes it. And in this video, I'm going to show why Dr. Carter is wrong in making the case for the Jeanson rates. And ironically, Dr. Carter is also going to show why Dr. Carter is wrong. So let's talk about that. The big picture for the rest of Dr. Carter's argument is no. Single generation pedigree mutation rates do not equal the phylogenetic substitution rates. I've explained this in detail and I've linked this video down below. In that video, I go through a whole bunch of reasons. The big one I focus on is how creationists actually cherry pick their pedigree mutation rates. And if you actually look at all of the data from pedigree studies, You actually don't see support for the fast rate that young Earth creationists need in order to make their math work. But I also allude to a bunch of other problems in that video, such as failing to weed out somatic mutations instead of just focusing on the germline, or misunderstanding the difference between a pedigree and a phylogeny. There's a whole bunch of errors that I run through fairly quickly in that video that give you a good overview of kind of the scope of the problem that creationists are dealing with. But in this video, we're going to focus specifically on what Dr. Carter said and highlight the problems with his argument specifically. The first problem with Dr. Carter's case that he builds here is the fact that he points out repeatedly that the Y chromosome and mitochondrial DNA are haploid, and that creates a problem for those particular bits of DNA experiencing purifying selection. The problem is that haploids experience purifying selection. Natural selection operates on haploids. Haploid, by the way, is when you have one copy of each locus in your genome, and diploid is when you have two of everything. So in the human genome, the autosomes, chromosomes 1 through 22, are diploid. You have two copies of each one. You have one from mom and one from dad. The Y chromosome and the mitochondrial DNA are necessarily haploid, with a few exceptions. You only have one of them. So typically you have a Y chromosome. If you're 46XY, you get one Y chromosome. Your mitochondria is derived from bacteria. The mitochondrial DNA, like all bacteria, is haploid. Now, the X chromosome could go either way. If you have one X chromosome, like if you're 46XY, then it acts like it's haploid. If you're 46XX, then it acts like it's diploid. So we're not going to worry about the X chromosome, but that could go either way. We're really just talking about Y chromosome and mitochondrial DNA. Now, Dr. Carter knows that haploids experience selection. He said it. And in this video right here, I'm going to show you a short clip where he describes how bacterial populations experience natural selection. Bacteria are, of course, haploid. Check it out.
Because they can be more affected by mutation, natural selection can see that mutation more clearly, and it can filter it out.
Now, I think what's actually going on here is Dr. Carter is alluding to the fact that if you're haploid, you don't do recombination, and that makes natural selection less efficient, right? He's talking about the concept of Muller's ratchet, which again is why I think he uses the phrase ratchet-like accumulation of mutations a couple of times throughout the video that I'm responding to. So we know that haploids experience selection. That's not actually the thing that he is saying. At least I don't think Dr. Carter is implying that if you're haploid, you can't experience natural selection. That would be a very silly thing to say. I don't think he's saying that. I think he is alluding to an argument that is related to the concept of Muller's ratchet. So let's get to problem number 2, which directly addresses that. The second problem with Dr. Carter's argument has to do with recombination and linkage. So let's just establish a bit of a background here in terms of what we're talking about. Take a look at this simple representation of a chromosome. The black bar here represents a chromosome, and each of these turquoise circles represents a specific mutation. Now, these mutations are all linked. They're located on the same physical piece of DNA. That's what linkage is. You have two different mutations that are on the same piece of DNA. They are linked. Now, if these mutations are not decoupled via recombination, you must select for or against all of them as a unit. If one of those mutations is beneficial, you're going to drag all the rest along with you. That's called genetic hitchhiking. If one of them is bad, the same thing happens. You got to get rid of all of them. You got to toss the baby out with the bathwater. You can get around this problem by having recombination, right? And you probably learned in biology class at some point about how the chromosomes line up side by side during meiosis and they swap little pieces. That's called homologous recombination. And that allows you to decouple mutations from each other so you can specifically select against the bad ones without necessarily selecting for or against any specific things that they're linked to. Linkage, in other words, reduces the efficacy of selection. It's harder to isolate the effects of harmful mutations linked with really important alleles, right? If you have a bad mutation over here, but on that same piece of DNA you have something you really need, You can't have selection against the bad thing because you'll lose the good thing along with it. This is the concept of Muller's ratchet, right? It's harder to get rid of the bad stuff when you're not recombining because it's linked to things you need. This does not mean that there is zero selection. It just means that the threshold for getting rid of something bad is higher. It has to be really bad in order to overcome the cost of getting rid of the stuff that's linked with it. It also does not mean that only harmful mutations are affected by selection. Linkage works both ways. Just like you can't get rid of a bad mutation if there's something really critical that's linked with it, the kind of counterpoint to that is also true. If you do get rid of a bad mutation, you get rid of all the other stuff that's linked along with it. So let's say that a really bad mutation happens on our hypothetical little chromosome here, right? Now this red dot indicates a very harmful mutation. How does selection against that mutation affect the frequency of all the other variants on that same chromosome? The harmful mutation is linked with all these neutral mutations. There's no recombination, so there's no way to isolate this mutation from its context, from its background, right? We can't select against that bad mutation in a way that's kind of indifferent to the things that it is linked to. So, the harmful mutation gets selected against, its frequency in the population will be reduced, and the frequency of all the linked neutral mutations will also be reduced. This concept is called background selection, and this matters for what we're talking about because this is what happens in the Y chromosome and the mitochondrial DNA. If you have a sufficiently harmful mutation, you're going to select out not just that mutation, but all the other variants that are associated with it because there's no recombination. This reduces the substitution rate relative to the mutation rate. So the question isn't how many of those sites are subject to selection. The question is, are there any sites that are sufficiently harmful to be subject to selection? And if the answer is yes, you're not just going to get rid of those, you're also going to reduce the frequency of the linked neutral variants through background selection. And again, the effect of that is to reduce the substitution rate relative to the mutation rate, which is the opposite of what creationists want to happen. And again, this is the case if there are sufficiently harmful mutations that they are selectable. And I don't think anybody disputes that that is the case. There are specifically like mitochondrial diseases, right? There are mutations in the mitochondria that are fatal to the mitochondria in which they occur and even to the person in which they occur. So obviously you can have selection against harmful mutations within the mitochondrial DNA. And the same is true for the Y chromosome, which means we're going to see a divergence between the mutation and substitution rates, in part due to background selection. Now this gets to the big question. Do we have direct evidence of selection? Because the third and final problem, and what we're going to spend the rest of this time talking about, is the fact that we do in fact have direct evidence of selection operating on the mitochondrial DNA and Y chromosome. The key question is this: are the mtDNA and Y chromosome affected by purifying selection, selection against new mutations? Right? A new mutation occurs, it's harmful, we select against it. We agree on the effect that that selection would have. We agree that it would cause a divergence between the mutation rate and the substitution rate. So does selection happen? That's what we have to establish. We can answer this question empirically for both the Y chromosome and the mitochondrial DNA, and that's what we're going to do. We're going to spend most of this time talking about the mitochondrial DNA because that's most of what Dr. Carter was talking about in his various videos that we're responding to here, but I will just briefly get the Y chromosome out of the way before we get to that. So let's get to the Y chromosome. I'm going to show you two papers here that show strong evidence of selection on the Y chromosome. So this paper is called Remarkably Little Variation in Proteins Encoded by the Y Chromosome Single Copy Genes Implying Effective Purifying Selection. And I'm just going to read this highlighted bit from the abstract of this paper because it summarizes what we're talking about here. Quote, There is remarkably little variation in X-degenerate protein sequences. Two chromosomes drawn at random differ on average by a single amino acid, with half of these differences arising from a single conservative ASP to GLU mutation that occurred about 50,000 years ago. Further analysis showed that nucleotide diversity and the proportion of variant sites are significantly lower for non-synonymous sites than for synonymous sites, introns, or pseudogenes. These differences imply that natural selection has operated effectively in preserving the amino acid sequences of the Y chromosome's X-degenerate proteins during the last approximately 100,000 years of human history. And here's a second paper on this same topic: natural selection reduced diversity on human Y chromosomes. Quote, "Here, using genome-wide analysis of X, Y, autosomal, and mitochondrial DNA in combination with extensive population genetic simulations, we show that low observed Y chromosome variability is not consistent with a purely neutral model. Instead, we show that models of purifying selection are consistent with observed Y chromosome diversity." And I just, before we move on, wanted to shout out this paper. I think the methodology that they used was pretty cool and pretty creative. This paper and the one I just showed you are linked down below, so I want to encourage you to check both of those out. But in particular, I really think the way the authors approached this study did a really cool job in terms of just how they kind of teased out the different factors here. So that is going to wrap it up for the Y chromosome. Like I said, we're going to spend most of our time here on the mitochondrial DNA. We could spend just as much time on the Y chromosome, but most of what Dr. Carter talked about was specifically regarding mitochondria, so that's what we are going to spend our time on as well. So let's talk about selection operating on mitochondrial DNA. This is a map of the mitochondrial genome. The mitochondrial DNA is relatively small. It contains 37 genes, and it's about 16,500 nucleotides. Notably, for the mitochondrial DNA, it has very small intergenic regions, and it is extremely gene dense. So you can look at this, and it's color-coded according to what it is, but you can see that there's a bunch of genes here. There's ribosomal RNA. There's also tRNA genes in here. It's very tightly constrained because it is very dense in protein coding and functional RNA encoding sequences. And importantly, all of this stuff is critical for survival, right? This has to do with like very basic metabolic processes. So these genes are extremely highly conserved. The sequence does not vary very much from person to person and even from species to species. It's very similar because it's highly constrained. That implies that there are very few neutral sites and strong purifying selection. In other words, if you mess with these sequences, it causes a problem and it gets selected against. Now, one thing Dr. Carter alluded to is the idea that evolutionists tend to argue that there's a lot of junk DNA in the genome, and therefore a lot of variation is going to be neutral, and he says you can't have it both ways when you're talking about the mutation rate versus the substitution rate. But Dr. Carter, we're talking specifically about the mitochondrial DNA here, and the mitochondrial DNA is emphatically not mostly junk. The mitochondrial genome is exactly what a gene-dense, highly functional, low junk DNA genome looks like, and in that regard, it's very different from the rest of the human DNA, so that argument does not apply here. We're not talking about the rest of the genome, which does have mostly junk DNA in it. The mitochondrial DNA is mostly not junk. In fact, it's mostly constrained at the sequence level. So let's see, if we dive into it, do we actually see evidence of strong purifying selection like we expect? The first thing we can look at to address this question is region-specific substitution rates within the mitochondrial genome, because certain regions are more constrained than others. You have coding regions. Within those coding regions, you have the first and second codon positions, and you have the wobble position, the third position. The third position should be less constrained than the first two. We also have hypervariable regions, HVS1 and HVS2. and more broadly speaking, the non-genic, the intergenic region called the control region. Now, this table is from a 2009 paper that I've linked down below, and this table, in the column I've highlighted, shows the substitution rates in terms of substitutions per site per year for these various regions. In yellow are the regions that are less constrained and have a much higher substitution rate, and in pink are the more constrained regions that have a slower substitution rate. You can see that the range of substitution rates for the different regions in the mitochondrial genome span a range of approximately 2 orders of magnitude, with the more highly constrained regions shown in pink having a much slower mutation accumulation rate than the less constrained regions shown in yellow. Now, unless Dr. Carter is suggesting that the baseline mutation rate is many times lower at the non-synonymous sites, at the ribosomal encoding sites, at the transfer RNA encoding sites, relative to the control region and the wobble position, which I don't think he's suggesting, because honestly, that would be a silly suggestion. I don't think anybody is suggesting that just inherently those specific sites experience a lower mutation rate. So I think we all agree on that, but as long as nobody is suggesting that, then purifying selection is operating on the mitochondrial DNA, specifically at the highly functionally important sites, the sites that if they change, they disrupt protein function or ribosome function or tRNA function. So right away, we have very strong evidence of purifying selection in the mitochondria. We also have direct observations of this divergence between the mutation rate and the substitution rate within the young Earth timeframe. Young Earth creationists like to claim that when we calculate the substitution rate, it requires an assumption of human-chimp common ancestry. This is wrong, and I've addressed this before. This is wrong for the very simple reason that we can actually calibrate these calculations by anchoring them at events with a known date. So like the date that specific islands were settled is a really good one, especially when those islands were settled recently within the young Earth timeframe. So for example, the human settlement of the Canary Islands and Vanuatu in the Pacific occurred within the young Earth timeline. And the divergence between the mainland populations and the island populations in each of those cases corroborate the phylogenetic mutation rate, even though these events are well documented and occur within the young Earth timeframe. We can also directly test the young Earth predictions on this, and I've done exactly that in this video, again, link down below. And when you test the young Earth predictions, they are wrong. And they're not just like a little bit wrong, they're extremely wrong. So I tested these predictions specifically for the Canary Islands, and here are the results. The predicted number of mutations using the young Earth mutation rates, taking that pedigree mutation rate and extrapolating it backwards in time infinitely, the predictions are that in the two populations, the Canary Islands and the mainland North African population, you expect 23 to 120 differences in their mitochondrial DNA. The actual observed number of differences is 2 to 4, depending on how you measure it. That means that the young Earth predictions, they're not off by like a little bit, they're off by a lot. At a minimum, they're off by a factor of 5.75, and at most, they're off by a factor of 60. Now this table right here, this is just a screenshot of the Excel spreadsheet where I did these calculations that I showed, and I walked through them in that video I just mentioned. So if you want all the details on this, you can check out that video. It's linked down below. But look, you don't have to take my word for it. There are plenty of publications that show and explain the robust evidence for purifying selection in the mitochondrial DNA. I'm just going to reference a couple of them right here, and all of these will be linked down below. From 2008, we have Purifying Selection of Mitochondrial DNA and its Implications for Understanding Evolution and Mitochondrial Disease. From 2018, we have Mitochondrial DNA Purifying Selection in Mammals and Invertebrates. And finally, from 2021, Avoiding Extinction, Recent Advances in Understanding Mechanisms of Mitochondrial DNA Purifying Selection in the Germline. And I especially like that last one because this is where we would expect a lot of the selection to operate. It's going to be within the female germline because remember, mitochondria are inherited through the maternal lineage. So a lot of this is going to happen within the germline for females. And if there's a mutation that is going to inhibit or prevent the egg cell from forming correctly, then that lineage within the germline is not going to persist and it's not going to be fertilized. And if it's fertilized, that embryo will not survive. So a lot of the selection is occurring in the female germline. And that last one really gets into the mechanisms of how that works. So strongly encourage everybody to check that out if you want the details on what's going on under the hood. Now, this does raise the question of just how much selection is going on here, because Dr. Rob Carter and others will say that their models can accommodate some level of selection. So say it's, instead of calculating a time to most recent common ancestor of 6,000 years, maybe it's 8,000 or 10,000 years. That's fine. There's different calculations for the age of the Earth among young Earth creationists, and they're not wedded necessarily to a 6,000 year number, right? So if it's only a little bit of selection, then it doesn't really matter. We can still deal with that. But then we can go back to this table and we can see just how big those differences are in terms of the effects of purifying selection in the highly constrained regions compared to the substitution rate in the less constrained regions. And as we saw a few minutes ago, those differences are 1 to 2 orders of magnitude. That's not going to lead to a slight change in the time to most recent common ancestor. That's going to lead to a big change, right? 6,000 years becomes 60,000 years at least. And that's really notable because that's consistent with the approximate consensus range for the mitochondrial most recent common ancestor of 60 to 200,000 years ago. There's one more paper that's relevant to this specific conversation that I want to bring everyone's attention to because it's a really unique and interesting illustration of the strength of selection operating on mitochondrial DNA. This was a paper published in 2007 in the journal Nucleic Acids Research, and the title of this paper is Mitochondrial Diversity Within Modern Human Populations. And I want to point out a really critical line of the abstract that shows the key finding of this paper. So here it is, quote, fully 84.1% of the mitochondrial genome was found to be invariant. Meaning in this sample from 2007, which admittedly is a little on the old side at this point, but I think the methodology was fine for what the data set that they were working with, 84% of the genome was identical across all of the samples. That is extremely strong evidence of purifying selection, unless you're going to claim that mutations are not occurring in the rest of the genome, and I don't think anybody is making that argument. So this is quite strong, direct evidence that harmful mutations are efficiently removed from the mitochondrial DNA. In his response to my video, Dr. Rob Carter did not mention this paper or engage with these findings, which I think is notable because this is a really important result that has implications for this debate about mutation rate and substitution rate and purifying selection in the mitochondrial DNA. And I think it's notable that he didn't bring it up because I know he knows about this paper. I know Dr. Carter is familiar with these findings because he wrote this paper. This is his paper. So I would be really interested to see how he addresses those data that, again, are extremely strong evidence of purifying selection in the mitochondrial DNA. So what is Dr. Carter's explanation? Well, after he released his response to my video, he released this video, The Saga of Mitochondrial Eve Continues. And in this video, he talks about 2 more recent papers with more extensive data sets that show basically the same thing that he described in his 2007 paper. So I'm going to show you a clip of this video. It's a little bit longer, but it's important, so we're going to go with it. We're going to watch this clip because he explains exactly why we're going to see this remarkable degree of consistency in the mitochondrial DNA. Check it out.
And then how constrained is the genome? And that's what these 2 new papers, the Lake et al. 2024 and the Larici et al. 2022, they looked at how much variation we see. Now, there are 56,000 human genomes. They showed that about 50% of the human mitochondria it never changes. They never saw any variation at that spot. Whoa, 50%. So half of it probably can't mutate or the individual dies. That's why you don't see it, because the mutations are happening, just you never see them. They also showed that of all the letter changes in the genome that would affect the protein, the amino acid in the protein, that you can have what's called a synonymous change or a non-synonymous change. A synonymous change is when you change the letter in the DNA, but it doesn't change the amino acid in the protein. Because as you know, when proteins are manufactured, they're manufactured from a messenger RNA. And there are codons in the messenger RNA. 3 letters in the messenger RNA is a codon, and each codon specifies which amino acid goes in the protein. But there are 64 possible combinations of 3 letters. You have 4 times 4 times 4. But there's only 20 or 21 amino acids. So most amino acids have more than one codon. So if you can mutate the codon, very often it doesn't change the amino acid. But out of all the possible synonymous changes, they only saw about half of them. They only saw about 10% of all the possible changes, mutations that would change an amino acid, which means most amino acid changes are bad. Yeah, it can happen, but they only saw 10% of them. And they only saw about 1% of all the possible mutations that would stop a protein earlier, or if you change the stop codon, make the protein go longer. So there's constraints there. But then in the Lake et al. Paper, it's amazing stuff. They showed that specific areas of proteins do not change, and they figured out why. They showed that specific areas of the RNAs don't change, and they showed why. In my not the splicing and dicing one. Oh, in the episode that I did called the spaghettification of irreducible complexity, I talked about in the middle of that episode how the transfer RNAs that have on the bottom an anticodon, the 3 letters that matches up with the codon in the on the RNA, the anticodon, and then the top they have an amino acid, and they're used in the ribosome to manufacture the proteins. But those transfer RNAs are heavily modified. They're modified sometimes 15, 20 times. So another protein will come and attach to it and change the letters of the RNA or stick something on it. And what Lake et al. Showed was that in the anticodon, you can't change the first 2 letters. The 3rd letter, that's more changeable, because when you have an amino acid that's 4 different codons, it's the 4th one, the wobble base, that can be different. It can be an A, C, G, or a T. So if you mutate that one, you keep the same amino acid. But if you change the other ones, you change the amino acid, that's probably bad for the organism. They showed that in the places that get changed on those transfer RNAs, those places are pretty much invariant. Oh, so the cell needs that letter there, so another protein can come and attach to it and make a chemical change to the RNA. Or the RNA doesn't function right, and the protein doesn't get made right. So there's a lot of constraint. There's a lot of evolutionary, if you want to call it that. There's a lot of evolutionary design systems, whatever you want to say, constraint. You can't just change anything. Only certain mutations are allowed.
So here is the critical quote from that video. This is what he said. This is the transcript of what he said. This is the critical line. Quote, "They showed that about 50% of the human mitochondria never changes. They never saw any variation at that spot." Whoa, 50%. So half of it probably can't mutate or the individual dies. That's why you don't see it, because the mutations are happening. You just never see them. This is the key thing. And I want to thank Dr. Carter for saying this, because this is exactly what I think as well. Because what Dr. Carter just described is purifying selection. 100%, there's no bones about it. Dr. Carter is describing purifying selection operating on the mitochondrial DNA. That is what he is describing when he made that statement. Remember this table that we saw a few minutes ago? This is exactly what he's talking about. This is the thing. He talks in that clip we just saw, he talks about how the wobble position has more variation than the 1st and 2nd codon positions because you can change the wobble position and not change the actual amino acid sequence of the protein. But if you change the first or second codon positions, you change the amino acid sequence, and that probably comes with a cost. He also talked about transfer RNA. tDNA is the DNA that codes for transfer RNA. Those sequences are really important. If you mess with those sequences, stuff doesn't work. This table with this variation in substitution rates is exactly what Dr. Carter just described. And the explanation that he just described, that we just heard him describe, that explains these differences in substitution rates in the different regions of the mitochondrial DNA, the explanation is purifying selection. He didn't use the words purifying selection, but he described purifying selection. 100% unambiguously, that's what Dr. Carter just said explains the remarkable consistency of the mitochondrial genome. So I want to thank Dr. Carter for his clarity on this issue. We agree. Extremely strong purifying selection operates on the mitochondrial genome. Most of that mitochondrial genome is highly constrained and experiences a substitution rate that is 1 to 2 orders of magnitude slower than the rate experienced by the less constrained regions, most notably the control region and the third codon positions within the genes. That's it. That's the end of the debate. The mitochondrial DNA experiences purifying selection. I agree with what Dr. Carter said, because that's what he just said. I don't have anything to add to that. We agree there's purifying selection, which means the substitution rate and the mutation rate are different, and that's the ballgame, folks. In conclusion, Dr. Carter claims what he said in that stream from way back in January was not contradictory with what Dr. Jensen says, that he wasn't talking about the mitochondrial DNA and Y chromosome at all. Now, I explained why I think that's not the case, but that's not really the main point I want to get across here. The point is that what he said about mutations being lost over time also applies to the mitochondrial DNA and Y chromosome. The big question is, does purifying selection affect the mitochondrial DNA and Y chromosome? And the answer is unambiguously yes. And Dr. Carter agrees with me. That's what we just watched him say in his own words, and you can go back and watch it again when you want. You can watch him again describe purifying selection in the mitochondrial DNA. It was completely unambiguous, and I'm not having this debate anymore. We agree purifying selection operates on the mitochondrial DNA and also the Y chromosome. And if selection happens, the young Earth timeline is wrong. They cannot accommodate a slower substitution rate in the time that they have, whether it's 6,000 years or 8,000 years or even as far back as 10,000 years. There's no way to accommodate the directly observed and calculated slow phylogenetic substitution rate in the young Earth timeline. But you have to acknowledge that that rate is correct if you acknowledge that selection occurs in the mitochondrial DNA and Y chromosome. And I want to emphasize this one more time. We just watched Dr. Carter describe how purifying selection operates in the mitochondrial DNA. So that is the end, folks. At least one professional young Earth creationist agrees with me on this question. There is selection in the mitochondrial DNA, therefore the young Earth time to most recent common ancestor calculations are wrong. I'm sure Dr. Carter disagrees with the therefore half of that statement, but let me just speak directly to Dr. Carter here. Dr. Carter, you're going to have a very difficult time convincing me that you disagree with the purifying selection occurs part of that statement, given what you said in the video that is, as I record this, the most recent video you have released. And that's where we're going to leave this, folks. Thank you all for watching. Please remember to subscribe to the channel if you're not already subscribed. Hit like, leave a comment, share this video, do all the, the algorithm-friendly things. If you are so inclined, if you appreciate what I do, you can become a channel member, and members get immediate access to pre-recorded videos like this instead of having to wait for the public release date. I'm also working on a couple other benefits that some people have talked to me about. I'm not quite there yet, but I'm working on it. So you'll get some cool perks if you are a channel member, especially if you are impatient and just want access to the videos immediately and not have to wait for every Wednesday. Thank you all again for watching, and don't get fooled.